Hey, it's Peter here with my FSHD. All right, sorry I've been kind of out for a little bit. When I got back from my trip to China and Vietnam, I came up with a respiratory issue. I've just been having trouble talking. So hopefully I can get through this okay. Because we're trying something a little bit different, so I'm not really going to be able to edit. So I wanted to talk about my trip to Vietnam and China and tell you, I'll do that in the next one. Also about the EpiCRISPR gene therapy trial that's coming up. But Avidity finally presented publicly their data from their phase 1/2 Fortitude trial. Is that what it's called? And I've gotten a lot of requests from people asking what do I think about it and such. And so shout out to my man Scott, who I ran into in Shanghai, got to hang out with. It was good to see you, Scott. Um, and had a good time with you and Eric and and Sherlock, um, and then of course Shireen has been asking, and it's just no number of people have asked. So I thought, well, let me get through. And then also I looked on Facebook, and people were saying I don't understand this presentation. I came up with the idea that I'm going to try to go through the presentation that Avidity put on. It's a public presentation, so I assume it's okay that I do this, and I will give you my take on what's going on. And my personal executive summary on my interpretation of the results are not as good as I had hoped for. Still positive, still better than way better than Fulcrum's results. I'm not entirely surprised. Briefly, that their 4 mg per kg is not any better than 2 mg per kg. I figured if it had been, then they would have released that data early. They released their 2 mg per kg data. You realize, remember, about halfway through the trial, and that was very encouraging. I was also surprised by some of the data that's missing, and also by the, I don't want to say magnitude of the changes or the differences, but anyway, we'll get through it. I'm still encouraged by it. I still give them an A. It's the best thing we got out there. But I will tell you right now at the front end, if I were an RNAi company, an siRNA company, an antisense company, a RNAi gene therapy company, I'd be saying we can do better, and I hopefully they can. But it's still pretty good. All right, let's get to that. I'm going to try to share my screen here. Let's see, I can do that right now. I hope what is being recorded is my screen being shared. Okay, so this was June 9th. Avidity had this investor talk, and I'm just going to go through. Now, I haven't listened to them give their version of the talk. It's just like when I review a paper or when I review a grant. I like to make my own interpretations on the data, and not have somebody lead me. I don't want to be, have, I don't want to say biased by what other people say. So I'm going to go through it myself. All right. Forward-looking statement. Now, briefly on the forward-looking statements, I would say I have no connection to Avidity whatsoever. I don't remember. I know they used our mouse model. I don't remember if we helped them or not. If we did, we didn't get paid. I honestly don't remember. And we helped so many companies just because everyone uses our mouse model. But I have no financial connection. They don't donate to our lab, so it's not like a foundation, right? They're not donating to us, and so no, just strictly Peter Jones speaking as Peter Jones, and not a representative of anything other than, I guess, my FSHD. All right. So one thing to note, I just kind of point this out looking out here. So what's a BLA, right? This is a biologics license application. Poised for 3, 6, in 12 months, starting year end 2025. I'm not sure I fully understand this. I thought it was 26, but at the end, we'll see how it is. But what I wanted to talk about is, again, listen, you'll see some masses that are going to be listed. You see a dose listed 2 mg per kg, 4 mg per kg. That's milligrams per kilogram of the person. Okay, that's how these things are dosed. But that dose is only based on this little siRNA right here. It does not count for the antibody here. And the antibody is a lot larger than the oligo or the siRNA and the linker. So should be known, That's what this thing is. It is an antibody conjugated siRNA. The siRNA is going to target the DUX4 mRNA for degradation. Okay. And the antibody is going to target the transferrin receptor, which is a protein on the outside of cells, not just muscle cells, all sorts of cells of the body, but it's going to help this get into the cell. Because that's always been historically one of the problems is getting the oligo into the cell in vivo. We can do it in lab in cell culture, but doing it in a person or a mouse or a monkey is different. And so this antibody part, just like the antibodies you have in your body, is going to recognize the receptor and lead to the internalization of the siRNA. And so this is actually the mass of what's being injected is much larger. And quite possibly, you're going to see that they have an upper limit on what they're able to do. It could be due to the antibody, could be due to the siRNA, could be due to the whole thing. They perhaps don't really even know. Now, ideally, what you would have is an antibody or a peptide or some sort of targeting molecule that would be specific for skeletal muscle for FSHD. Now, you got to remember all of these companies, most of these companies are platform companies, right? They need technology that's going to, they're not really FSHD companies, right? So FSHD is one of the diseases they go after. Typically, they also go after Duchenne, muscular dystrophy, and myotonic in their neuromuscular space, but they often have liver diseases, blood diseases, nerve diseases, all sorts of things that they're going after. And so having an antibody that links to the siRNA that binds to all sorts of different types of cells is actually good for a platform company. But if you really only care about FSHD, you really only need something that goes into skeletal muscle. Now, that's easier said than done. But you know what? I'm willing to bet companies out there are going to figure it out if they haven't already. And what does that mean? That because now it could be safer, and you could also use a lower dose to get the same amount into muscle. Because remember, this is going to bind many different cell types. So these oligo is going to go into all these different cells that do not express DUX4. Okay, so this is where you're dosing. Can you get enough into the skeletal muscle cells that express DUX4? While all the other cells of the body are soaking this up. Okay, and this is why you want to have a higher dose for this type of thing. And so, like one of the concerns I have is that they don't. They seem to have reached a dose limit, and the question will be then: is that good enough? And I'm honestly, in my opinion, it's better than anything we got. It's better than nothing. Um, and I hope we can do better. That's what I'm going to tell you. All right, so this is their okay, they're talking about they're going to have a new study called Forward, phase 3, 200 participants, 18-month trial, has already been initiated. And it's interesting because I was unable to find this on clinicaltrials.gov. One-to-one randomized double-blind placebo-controlled. So 100 people are going to get placebo, and 100 people are going to get the Delbrax, okay, the siRNA. Okay, so that's something to keep in mind when if you're thinking about getting in this trial or if you're in this trial. Okay, randomized. You can't ask the doctor, "Hey, give me the good stuff." I mean, you can, but they don't know because it's double blind. The doctor doesn't know if they're giving you the placebo or not. Okay, so that's what that means. That's the phase 3 going forward. Now the key is we're going to talk about here is this the phase 1/2, and just remember that the thing that's important, what they're actually assaying in a phase 1/2 is safety and tolerability and a dose escalation. Okay. And what they found, that's their primary endpoints. Is this safe? Do you have any adverse events? Has very favorable safety and tolerability. And then the secondary endpoints, they're going to say, do you see any efficacy? The muscles get better or whatever. Okay. And biomarkers. Now, phase 3 is about efficacy. That's why there's many more participants, and that's going to be properly powered to see, does this actually show real benefit in patients? Okay, so you get an idea of efficacy from the secondary endpoints in phase 1/2, but there's just not that many people in it. And you will really get confirmation, well, hopefully, of efficacy in the phase 3. Okay, so phase 2, Fortitude. And then, of course, they had this biomarker study, which was one of the exciting things. I got to tell you, I was surprised at the biomarker. We'll talk about that when it comes up. I believe there's better biomarkers out there as well. That's why I was surprised. It's like, wow, they didn't use the good one. I shouldn't say that, sorry. But they use one that's okay, but it's no better than CK. But there was an additional arm where they're looking at the circulating biomarker that correlates with DUX4 expression. And remember how this is supposed to work. In FSHD, your skeletal muscles are making, just a very small percentage of your skeletal muscles are making DUX4 mRNA, which turns to DUX4 protein, which then is going to lead to a pathogenic cascade, right? The siRNA is going to bind the DUX4 mRNA and lead to its degradation. Therefore, you do not make DUX4 protein, okay? And that's what's going to go on. Now, because DUX4 is only expressed in skeletal muscles, they had to take biopsies in the phase 1/2. You have to get a biopsy before you're on the drug, biopsy after you're on the drug, and then sometimes even a third biopsy. Okay, this sucks. If you've had a biopsy, it sucks. You don't want to have to do it. So ideally, we wanted a circulating biomarker. Well, circulating serum creatine kinase has always been a biomarker for most neuromuscular diseases. It's an enzyme that escapes from muscle cells when they degrade. And now the issue is, there are a number of things like exercise can affect creatine kinase levels. Okay, and so CK is not a tremendously reliable in FSHD, or hasn't been thought to be. Levels are much lower in FSHD than they are in other neuromusculars like Duchenne. Duchenne is going to be 200, 2,000, 3,000, numbers, levels of CK, where in FSHD there might be just, 600, 700. So it's a lower window of SA. And so the idea was to find a new circulating biomarker. And so there was this additional, when the initial phase 1/2 data showed that they were actually knocking down DUX4 regulated genes, which was great because Fulcrum never showed that. So they're actually showing that it's working. They said, okay, let's start a biomarker study, a whole arm, and because they were looking at circulating biomarkers, and they found a biomarker that seemed to track with the small number of patients that were having DUX4 reduction. So they started this new arm, the biomarker arm, with more people to really validate this. And they're going to show that they were able to validate the biomarker. But again, it's not a whole lot better than serum creatine kinase. It's kind of funny because I was talking with another company, totally new, different company, about this a while ago. People are trying to say, Jones, do you know what the biomarkers? And the truth is, even though Stephen Tapscott's a good friend of ours, a collaborator, he never told me what his biomarker was for the Avidity. And so I didn't know. But like I said, well, the data just looks the same as CK that they put out there. And I was like, oh, yeah, but I was sure it's going to be better. It's not. It's different. But it might be more reliable to DUX4, though, because it's not going to be variable due to other non-DUX4 related events. So that's maybe where it's going to be better. Okay, but that's what we're going to talk about. All right, so I'm going so long. Most of the talking done was by Jeff Statlin. You can't do better than Jeff. Dr. Statlin, University of Kansas Medical Center. I would say now that Dr. Twill is retired, that Jeff is the number one. Sorry, I don't want to offend anybody. Just fantastic. Great. If you have a chance and you're in the area, that's definitely a medical center you'd want to go to, FSHD. I recommend to a lot of people to go there. He runs with Nick Johnson, they run the FSHD CTRN, which is the Clinical Trial Research Network. But Jeff's going to, I'm actually not going to listen to Jeff. See what he has to say. Okay, I don't really care. Okay, so this is the trial. Okay, so it's a dose escalation. What that means, they tried 2 doses, a 2 mg per kg and a 4 mg per kg. Now, you got to remember, these companies have they're platform companies. So they've done trials in other diseases. So they've done a myotonic trial where they did, it was either 6 or 8 mg per kg. And they had a lot of adverse events. And so they knew that 4 mg per kg is going to be probably the highest that they're able to go. So they did a 2 and a 4. Okay, and so if I understand this correctly, it's going to be dosed every 13 weeks, basically quarterly. So after you get your first dose, 6 weeks later you get your second dose, and then 13 weeks later you're going to get, and then 13 weeks on, you're going to be getting your siRNA dose every 13 weeks. And it's either 2 mg per, it's the 2 mg per kg first dose was 1 mg per kg. Interesting. Okay, that's how they did it. And then afterwards, once this has been completed in the 12-month study, open label extension. So it doesn't matter whether you're on the placebo or the drug, you get the good stuff in the open label extension, called open label, because you know you're on the drug. And you're going to get now you're going to get this every 6 weeks. So the open label extension is going to be dosing every 6 weeks. Okay, so that's how it's going to go. You start, you're going to get a muscle biopsy before you start the trial and a muscle biopsy 4 months after being in the trial. So there's only 2 muscle biopsies in this, and then you're going to get it whether you're on placebo or not. And again, the primary design, the safety and tolerability, right, of Delbrax, pharmacokinetics, and then they're going to look at some of the secondary endpoints. Okay, these are kind of exploratory things, so they can better design the phase 3 trial, reachable workspace, quantitative muscle testing, timed up and go, the 10-minute walk run, or sorry, the 10-meter walk and run test. They talk to the patients and the clinicians about how they're doing better. Are they doing better than biomarkers? Okay, so this is what's going on. This just tells you that they had 13 on the placebo, 8 were on the 2 mg per kg, and 18 were on the 4 mg per kg. I think they thought the 4 mg per kg was going to be pretty good. They're pretty similar groups. They're running about a 5 to 6 repeat, D4, Z4. And you'll notice there's no kids, right? Age of first symptoms, going to be mid-20s. And I guess they had some people as, well, anyway, there were no kids in this trial. Although, as I understand it, they're opening it up to at least down to 16 for the phase 3. All right, so this is the primary outcome they're going to be looking at. This is going to be looking at adverse events, right, and serious adverse events. And really, during the trial, there really, kind of weren't any that were related, any serious ones. In the open label extension, I guess there were a couple, but not related to the drug, right? You can have a serious adverse event, and then you find out and you investigate, and you find out it's actually got nothing to do with the drug. Okay, so related to drug, they had 3 on the placebo. Well, that's kind of bizarre that the placebo, right? 6, 11, and 9 in the open label relating to the drug. Okay, but again, these were just any adverse events, mild or moderate, nothing serious. Fatigue, decreased hemoglobin, platelet count can be one related severe non-serious herpes. Okay, yes, herpes is not, you didn't get her. Maybe you're feeling really good and you went out and you weren't too careful and they ended up with a herpes. Zoster infection. Okay, be careful out there, folks. Don't get too excited about getting on into a clinical trial. Okay, so great safety profile overall. Okay, so let's look at the top line data. Dr. Statland presented it, but now you got me going over my idea. Boy, I always see these numbers on FSHD, and to me, they're low. I'm going to say there's about 120,000 people in the U.S., just in the U.S., that are going to have FSHD. We're just finding all sorts of people with mild FSHD, asymptomatic, which may become symptomatic later in life. There's a lot of people with pain that don't know where it's coming from, fatigue. A lot of these characteristics we're finding when people eventually find us for our research testing, we're finding that they have the epigenetic consistent with FSHD. Now, in the end, you want this number to be down to 0. You want this number to go up. But the reality is, I think there's around 120,000 in the US, about over 1 million worldwide. Okay. And again, it's aberrant DUX4 expression is the cause. Okay, what matters to patients? Now we tell you it's interesting. We pain. I find this kind of interesting because we've asked people. I've always felt that pain has been the #1 complaint that people have with FSHD, but only 2% says that's the most meaningful thing. Loss of muscle function. I guess if you maintain muscle function, but you'll put up with the pain, I guess is what this is saying. So you really want to stop the loss of muscle function, right? And that's a lot of times, I think that's actually what the data is going to show us, is they're stopping the loss, not necessarily in this short amount of time regaining strength, but maybe some people are. It probably, when I talk to people online, I always say it probably depends on where you are, right? And so it's, the more affected you are, the less likely you probably will regain strength and activity, the less effect. So this is why, in other My FSHD podcasts, I always say, hey, let's make sure that's why we work with Tamara Gottlieb and Wayne Nesbitt and Emily Shaw. is that we want to make sure that you're in the FSHD supplements, nutrition, and peer support, right? A Facebook group. We want to make sure that you're in the best possible health you can be when these therapies are coming, because that's going to be your best chance. And it was interesting. I put this on my phone. I found this on my phone. I got this email to me on just something else random, from Hippocrates. The patient must combat the disease along with the physician, right? And what that means is you got to do your part. And so I think the drug will be able to do the part of stopping progression. And that's at least what I'm going to show you that the data looks like it's showing to me. Regaining it, well, you can do some of that now, even if you're not in the trial. Put yourself in the better physical condition, tune the car up, and let's get going. All right, so improved functional mobility in Delbrax treated participants compared to placebo. Okay, so looking down here, they do an interesting thing here. They actually tend to combine their groups. And so this, see down here, it says 2 mg per kg and 4 mg per kg arms are pooled because they didn't really see a difference, right? And there's 18 people in the 4 and 8 in the 2. And so you put them together, that gets you your 26, if I can do math, treated people, so they're pooling them. And this is the 10 meter walk run test. And what I get out of this, the way I read this is, the change in the number of seconds it takes an individual to complete the 10-meter walk run test. I've never done it myself, but I can imagine what it might actually be. And so 0 would be that, again, this, you're going to say, you're going to get your baseline before you're on the drug, and then you're going to get this 12 months after you've been on the drug or placebo. And if it's 0, that would mean that you do this test in the same amount of time. If it takes you 20 seconds to do it, it still takes you 20 seconds to do it. That would be 0. And what they're finding is that if you people on the placebo, on average, it took them 2.5 more seconds to do this. They're slower. They're getting worse. Okay, and that with a 1, with pretty big error bars here. And if you were on the drug, those are the people that were on the Delbrax, they're fractions of a second. They're pretty much stayed the same. So this would suggest they didn't lose any functionality. They maintained whatever mobility they had by being on the drug compared with the people that were on placebo, which lost some mobility over time. We know it's slowly progressing. There's no statistics on this. Oh, the 24 right there. There you go. 24 and 11. That tells you what it is. And so, is this statistically significant? Yeah, I don't know. It's doesn't really matter. What the key is that on the drug, you're improving. Now, when they say improving, you're maintaining functional mobility based on this test. All right, so let's look at this one. Delbrax compared to placebo on the timed up and go. You sit and remember. So one of the inclusion criteria for this is that you need to be able to get up out of a chair without assistance. That's why, because they use this timed up and go. Sitting in a chair, you get up, and then you're going to go around a cone and walk back. So timed up and go looks like you go 3 meters each way, so about 6 meters. Again, they're going to pool the treatment arms, 2 mg and 4 mg per kg. Again, that's one of the disappointing things, is that the 4 mg per kg was not better than the 2 mg per kg, which again was not surprising because they did not release that data early. They released the 2 mg per kg data, and then we never saw the 4 mg per kg data. And that's fine. There's nothing wrong with doing that, but it told me that it probably wasn't going to be better. So I was not surprised. And again, you see the same thing. Virtually the people that were on the on Delbrax saw virtually no change in the time that it took them to do this. And the people on placebo, some people it took them, almost 6 seconds longer to do this. But other people, well, it only took them 1 second longer. But the key is, it looks like everybody took longer. And again, it's a pretty tight group here. That's actually strikingly tight. That it tells me that most everybody stayed the same. Again, maintaining your functional mobility based on this assay. All right, let's look at the next one. Improve strength. Okay, now you're going to look at elbow flexor, shoulder, ankle, dorsiflex, knee, basically quantitative muscle testing. Okay, And what's going on here? So you're going to get a strength number, and based on some probably cumulative score, I'm going to say those that are on the on Delbrax. Basically, again, it doesn't look like they really got stronger in general, but they didn't get weaker. On placebo, got weaker. Okay, this is what it looks like. Now it would be interesting to see all 12 of these put out, and also now we're up to 26. So everybody did this one. Previously it was 24. Um, Whatever, it doesn't matter. So that's, again, maintain. We said we can stop the destruction, stop muscle loss, and everybody said that they'd be pretty, well, not everybody, 60% of y'all said you'd be pretty happy with that. So there you go. Let's stop the destruction first. All right, improved upper limb function in Delbrax treated patients compared to placebo. This is reachable workspace, which I hate. The reason I hate this is you can game the system, you can game this, you can practice this, you can work this out. And when I talk to a lot of patients that have trouble reaching up or upper, they'll talk about having good days and bad days. They said that some days they just don't feel like they can do it. Other days they can do it. Or you can kind of work yourself to be able to do this. You can kind of compensate to be able to do. So it's not my favorite assay. And I think what you see is that's why there's so much variability here. So reachable workspace and relative surface area covered. And they're going to look at Q1 and Q3. So again, you need to do, that's why the inclusion criteria, you need to be able to get your arms up, right, in order to do this assay. If you can't raise your arms up, they're not going to want you in the trial because you can't actually participate in this, right? Although Q2 and Q4 would be interesting to see, but they're going to look at Q1 and Q3, and they're going to give you a weight when you do this. So you're carrying a weight. It's not just without a weight. And you see, and now they're going to, so this is what's really funny. I remember Dan Perez asked me, Ben Perez just pressed and pressed the reachable workspace. One guy said, so what am I getting here? Are you telling me that you got, what, a quarter of an inch more functionality? I mean, so what does this mean? 0.02, okay. Honestly, I'm not entirely sure what that means, but if when you make a graph and you want to show something, pay attention to what's on the axes, because you might say, wow, that's a huge difference. The reality is this is less than 0.02 and this is negative 0.02. The difference between this and this is 0.04 whatever's reachable workspace is. Now, if it was 0.04 yards, that would be one thing, but I'm willing to bet it's like 0.04 inches or centimeters even. So relative surface area, honestly, I don't know. On a certain level, it doesn't matter. The point is that people that were on Delbrax were able to cover, even though it's a little bit, they were able to cover more, and the people on placebo covered a little bit less. It suggests, again, it indicates that being on the Delbrax, you actually improve. I would say more than anything, you didn't lose functionality. That may be the most important. These people are losing a little bit of it. These people are, it suggests their gain. I mean, the numbers are so small, I'm not sure if this is a meaningful gain or not, to tell you the truth, and a lot of variability. All right, functional mobility in Delbrax treated participants relative placebo exceeding MCID at both doses. I can't tell, I just kind of breezed through what the. Okay, MCID, minimal clinically important differences from the Resolve study. Now, you'll remember that Jeff Statland is in charge of the Resolve study, right? So this is a big, it's like 400 people in this study, I believe, where a natural history study that's being carried on multiple sites all around the world, actually. And studying, this is, studying just, the question is, how fast does disease progress in somebody, right? This is a 12-month trial. Should we even expect to see a change in 12 months? Some metrics, maybe you don't see any change. It's just natural, right? Or how much variability. And so this is why it's been very important to be in these trials. The natural history study trials kind of suck, right? You're like, I'm not on a drug, I'm not on a treatment, there's no intervention. But it's really important to understand if the interventions are working. So anybody who has been in the Resolve trial, study, thank you so much. It's been really great to have you in there, especially those of you who gave bio. I think that's a MOVE study as well. So the MOVE study also tells you these things. That's, I think I'm thinking of the MOVE study, and that Resolve was probably the Fulcrum study. No, that was Redux. Sorry. See, this is why I can't do this live. Anyway, natural history studies that let you them know what is a clinically important difference. Okay, that's basically what comes out. And what they're going to basically show is 2 mg per kg and 4 mg per kg. There's really no difference. Whatever this point here is here, 1 second in the timed up and go. So if you can do something 1 second faster, or if you're doing something 1 second slower, then that is a clinically important difference. 1 second in this assay is meaningful. You are actually slower or you are actually faster, right? And they're going to say that this by basically that's 1 second. So they're going to say 4 seconds here. Looks like 3.5 seconds. That is clinically meaningful. For the 10 meter walk run test, basically 1 second again is clinically meaningful. This is a cleaning. So even though these changes are small, I say it's only 1 second. For these assays, 1 second does actually matter. And so that's actually really important. And now the other key is it didn't matter whether you're on the 2 mg per kg or the 4 mg per kg. You didn't see any difference. So they topped out on probably what they're able to get into the cells. All right, same thing for reachable workspace. In this one, the 2 mg per kg is actually better than the 4 mg per kg. But look at these error bars all over the place. So I don't know. Same for the quantitative muscle testing. And then now this is a quality of life index where they kind of interview people, as I understand correctly, talking about upper extremities, talking about sleep disturbance, physical function, fatigue. Are you more tired, less tired? Do you feel like you're functioning better? And again, in every case, the people that we're not getting, people, remember, people are blinded. The physicians are blinded. Everyone is blinded to whether or not they are on placebo. The person that's doing the analysis, the interviewing, it doesn't know of the person. They don't know, oh, here's someone on drug coming in. They have no idea. It's double blinded, randomized. So all these metrics are done with the patient, the doctor, the assessor, the person putting all the data together. Nobody knows until it gets unblinded at the very end. And you can see in every case, all these metrics, being on Delbrax was better than not being on Delbrax, even though it's small differences. I don't really know what a sleep disturbance difference metric is, but whatever, it's better. How do you feel? Pain? Well, now you got to also remember some people that are on placebo think they're on drugs, so they might actually. So this is where the patient-reported outcomes are where you have the more variability. Strength is strength. Although you can try harder to some point, you can go as fast as you can to some point. But if you say, "Hey, how are you feeling?" Well, everybody that's in a clinical trial is feeling better because they're in a clinical trial and not placebo. So you can see that it's there's a little overlap here, but it's still striking that everybody that's on Delbrax really reported that they're doing better. The patient felt like they're doing better. So overall, this shows that compared to placebo, I would agree with this. Improved. Boy, is this statistically significant? I don't know. They didn't run statistics on it. I would say that improved is a dangerous word. Improved, they would have better functional mobility than people that run placebo. I guess you can use improved. I don't know. Improved tells me that they got better. Maybe they got slight. To me, what I got out of it is they didn't really get worse, and maybe they got a little bit better. Improved muscle strength. Again, same sort of thing. They didn't really lose muscle strength. Improved quality of life. It did appear to be improved quality of life. Favorable long-term safety and tolerability. Agree with that. And then now we're going to talk about the biomarker trial, okay, and see what's going on with that. Okay, Dr. Flanagan, we're not going to listen to him either. So this thing they trademarked it, they're calling it CDUX, a game-changing circulating biomarker with the potential to accelerate Delbrax for people living with FSHD. Not sure it's game-changing. At the end of the day, but it's good to have multiple circulating biomarkers to track what DUX4. Okay, so CDUX is elevated. This is what they're going to say, 6 to 9 fold. When you have FSHD compared to controls, and they elevate with worsening disease. Okay, CDUX and CK. This is creatine kinase. Okay, that's one. They are statistically significant reduction in treated patients. CK is an indicator of muscle damage, and the crossover is pretty cool. We'll talk about that. And a biomarker for accelerated approval. This maybe is where I get out of my depth. I'm not sure. I mean, CK looks as good as CDUX, but it's a secondary endpoint in the Fortitude biomarker cohort. And the FDA has actually, I guess, said that the accelerated approval path is open, which is good news. Again, and not stunning results. One of my reasons, my initial excitement, well, and I'm going to tell you something, they're missing something here too. We're going to talk about this before we go to the biomarkers. What's missing? My official time, I'm going to pull up this other one here. Remember this study here? This was the early stuff that they put out, right? And remember here, let's see what we got. They had a little bit different, right? Here we had SIR, okay, that's pharmacokinetics. They're going to look at DUX4 regulated genes. You'll remember in the Fulcrum trial, right, that that was the Redux 4 trial. They had Ludex, TRIM43, KHDC1L, and MBD3. Right? This is the Avidity gene panel. Fulcrum had more genes that they looked at, and then there's even more genes you can look at, gene baskets, as we're going to call them. DUX4 is a transcription factor, meaning it functions to turn on genes, and actually it turns off a few genes too, and so as many transcription factors do. And so you can measure DUX4 activity by looking at gene expression, right? And you can actually look at DUX4 directly. That's what our lab always does. We look at gene expression for DUX4 and its regulated genes. And since the siRNA is targeting DUX4 mRNA, no DUX4 protein, you should see a return to a healthy transcriptome in DUX4, okay? And so they showed that here, right? This is what was really exciting to me. This is why they're taking your biopsies, right? They're taking biopsies so they can look at expression. And here, like it was just amazing, right? So like the 2 mg per kg, on average, a 53% reduction in the Avidity panel, which is Ludex, TRIM43, and KHDC1L, okay? And I thought, well, that's really great. And some people actually, as even over 70%, over 75% reduction in DUX4 target genes. Fulcrum never saw anything like this, right? And everybody went down, which is great. Everybody that they showed went down. So this says that the siRNA is reducing the target, right? Because you got to remember, you can get improved functionality without hitting the DUX4 target. That's a kind of, I think, what Fulcrum is doing. If you affect immune inflammation or immune function, or change the kinetics of muscle independent of DUX4, you could see temporary improvement in some of these muscle metrics. So I understand the FDA and patients are more interested in functional improvement, increases in strength. This is what I want to see. I want to see that you've knocked down the target. And because I believe if you do shut down DUX4, you will get the improved muscle function and strength, and maintain your strength, and maybe even gain your strength again, depending on how healthy you are. And the reasons, because I don't know how long it's going to take. Is it going to take 12 months, 2 years, 3 years, 4 years? I don't know how it's going to take, but if you can shut down DUX4, stop the destruction, you will catch up. You will be doing better, right? And so I want to know that it's due to DUX4, because if it's independent of DUX4, as I believe the small changes that they saw in the Fulcrum trial were independent of shutting down DUX4. They were affecting inflammation or some other DUX4 independent pathways. You could get a false kind of result. It's maybe not a therapy. So I love this. This is why I was so excited, right? And then we saw the Redux 4 panel. This is the Fulcrum panel, right? That's actually looking at like 6 genes, a few more are thrown on there. The 41 gene panel, 60. So this was great to me to see all of this. And I don't see any of this in this current set. Now, maybe they're going to show this in a subsequent study. They took the biopsies. They have the data. But see, look at this. This is what we're talking about. We're talking about going to a healthy transcriptome, right? The blue in FSHD becomes red, and the red becomes blue, right? The FSHD downregulated transcripts become upregulated, and the FSHD upregulated transcripts become downregulated. This is restoring your FSHD muscle to a healthy transcriptional state, a healthy gene expression state. This is what the drug is designed to do. Target DUX4, get rid of DUX4, return the transcriptome to normal, to a healthy state, and then now you don't have these cascades of pathology starting. Okay, and so that's what I actually really like. And there was none of that in this presentation. Now maybe they're going to present that at the FSHD IRC. Maybe that'll be in their next presentation. They have the data, but I was kind of frustrated that I didn't see any of that. And I'm now keeping in mind, because they should have, again, they took the biopsies, as I understand it, they took the biopsies. Now maybe they explained this in the talk, and you guys all know this, and I missed it, but anyway, my understanding is they took the biopsies. Then you have but you have the circulating biomarker. So for the phase 3, I'm glad that they don't need to take biopsies. I want to see the data that they already that they have, but I'm glad that they have a circulating biomarker. And interestingly, that circulating biomarker, the CDUX, is actually one of the 4 genes that they were looking at in your skeletal muscle. Okay, and so that is this KHDCL1. I gotta tell you, I did not see this coming. I was pretty sure it was going to be one of several other genes that I'm aware of, which may still come out in some future study, and maybe not. I bet they do. But this is, it's just a weird name, K homology domain containing like protein, whatever, RNA binding protein. Germ cell biology makes sense that it's overexpressed in FSHD because DUX4 is kind of a developmental gene. It is directly upregulated by DUX4. It is definitely in my FSHD derived. I'm not sure if it's myoblasts. Significantly elevated people with FSHD and correlated disease severity. And again, this is coming out of Stephen Tapscott's lab at Fred Hutch Cancer Center, one of the best labs in the world in FSHD, molecular biology, gene expression, muscle biology, you name it. They're the best. It's really great group. All right, so here's the data. Now here in black is going to be the levels of, they're going to call it CDUX, I guess, and over the course of the testing on placebo, right? And the orange is going to be on Delbrax. So you get on the Delbrax and it goes down pretty fast. And it does kind of bottom out at about a 40% change. Kind of comes back a little bit. It's kind of interesting that it's kind of coming back here at 12 months. 20%, you can see the range, it appears to be down. And now, interestingly, there are statistics on this, right? So, wait, I'm assuming, yeah, statistical significance, 0.00, pretty significant. Notice there were no statistics on the other stuff. People tend to show statistics when it's statistically significant changes. You see this one, for example, this is not a statistically significant change, but this is. Probably the people in placebo, it's going way up. Here, it's so the difference between these groups is actually what you're really looking at. What are we looking at over here? What are they different? Oh, this is a creatine kinase, CK, right? And actually, look at the CK. It's pretty much the same. It's not going down to 40%, but it goes down and levels off. And you could make the case, and they're all significant. They're looking at CK, so that's going to be their primary marker here in the phase 3. This will be the secondary. That's reasonable. I like it. I love this though. I love when you do the crossover, right? So people that were on placebo come in here, right? Then you get on the open label extension, meaning now all of a sudden everybody's on the drug. And you see the people that had been on Delbrax maintain good levels, low levels of the circulating biomarker. But the people that were on placebo that had high levels, now it suddenly drops right down pretty quickly. And that's good. And that's fantastic. That really shows that it is due to the function of the drug. And this is exactly what you want to see. This is fantastic. And so very happy to see this. So circulating biomarker, hopefully this means they don't need to take any muscle biopsies from anybody anymore. I love it. Okay, so although I gotta tell you, I don't know, maybe it's just because I just conceptually was like, well, it's gonna be better than CK. Oh, whatever. It's pretty good. So direct target, sustained levels, whatever. Okay, so there we go. Registrational studies. They did this code, whatever. Key Fortitude cohort. going on there? Wait, what is this? This is the biomarker cohort. Okay, had 51 people in it. They also did some other endpoints, key biomarker endpoints. They did not need to take any biopsies. So the whole thing between the two, there's 90 people, and that'll be in the regulatory package. Okay, so there's more data coming. FDA is in alignment. That's good news with the design of their forward study. And it's going to be North America, Europe, and Japan. Interesting. Not Australia, not Brazil, not South America. We'll have to find, and evidently not most countries in Asia. Interesting. Okay, it's going to be an 18-month controlled study, 1-to-1 randomization. Again, remember the dose listed is siRNA, and again, that's what I was getting at in the very beginning. You know, most of the mass of this is actually the antibody conjugate, and then you get an open label extension. Fantastic. Confirmatory trial design. Now, the one thing I would say, cautious. Most failures in drug development are in phase 3. Okay. Now, Fulcrum failed in phase 3. To me, it was fully predicted by phase 1, 2. No surprise that they failed. I'll be surprised if this fails. I'm encouraged by this. When I say that why am I not jumping out of my chair? Well, because I was hoping for better. I really was surprised that the 4 mg per kg was not better than the 2. They seem to have topped out. I honestly, I look at these things. So they didn't fully stop progression. They slowed progression, and that looks like they slowed progression. Everybody didn't fully stop it, and I didn't see much gain over 12 months. Now, maybe that's a tough ask in FSH. We don't really know how much can you gain, but there you go. And I think that the technology works, but if I were a company, if I had a company or involved with a company in siRNA or antisense or any of these other things, I'd be encouraged to say, hey, technology works. It can be done. They got a stake in the sand, but I think we can do better. I think you want to have something, you got to start somewhere. This will be the first one, but I do believe better stuff is coming. And again, don't take that in any way to be negative on Avidity at all. It's maybe I'm wrong. Maybe this will be the best we can do. Maybe I'm just being hopeful. Although I guess human data is really the key. That's the gold standard. Let's see what we got here. I think one thing that I saw, what's the last thing I wanted to see? Somewhere I had written down that it was going to be, I have it written down that it's going to be 245 sites worldwide. Does that say that in here somewhere? Where did I see that it's going to be at 45 sites? Because again, that only means there's going to be 4 to 5 people per site. Okay, so if you don't get in the trial, it's not the end of the world. We haven't seen Arrowhead's data at all yet. They have a phase 1, 2, same technology. They're probably slightly different target, same antibody. We know that Dyne is going to be getting into, well, we hope that Dyne's going to be getting in the clinic soon. That's going to be a little bit different. Myricule is coming right along. I love the guys in Myricule. Anthony Sala, really good group there, is leading. They're going to have slightly different technology. You know what we say, the devil's in the details, and the devil is in the details. And there's a lot of details here. You can have different sequences that are going to, so the DUX4 mRNA is like 1,500 bases of DNA. Different companies are going to target different regions. Some might be better than others for knocking it down. They're going to have different linkages. They're going to have different targeting molecules, although a lot of people currently are using the transferrin receptor because they're platform companies. Well, and I bet there are some companies doing other technology to more specifically target skeletal muscle. I hope that's the case. I have heard rumors out there. I mean, actually, companies that have nothing to do with FSHD, right? This is actually a lot of the search for skeletal muscle specific, there's probably going to be more striated muscle, right? Cardiac will be included probably, but to find something more specific. And then it can also come down to manufacturing cost and also delivery. Remember, this is an IV injection every 6 weeks. Every 6 weeks, you got to get an IV injection. Wouldn't it be nice to get a sub-Q injection, right? Just under the skin. Understand, I guarantee you the companies are working on that. Different ways to deliver it. And then every 6 weeks, that's a long time. What other metrics could you change? Maybe every 6 months. Wouldn't that be much better? That could get the cost. Getting it. So there's a lot of things that can still change. This is the first point. To me, it's positive. Actually, everything is positive on this. Everything I saw, I liked. It's positive. I just like to be more positive. But so don't please don't take anything negative out of this. I guess I hope we can. I'm sure that we can do. Hey, you know what? They may even do better. They may even have a next generation thing out there that maybe is even better. So it's interesting. I was talking to my friend Mick. I'm the Mick Hitchcock PhD endowed chair of medical biochemistry at UNR. And Mick, he worked for Gilead for 25 years, took a lot of their key programs all the way through. And he's like, you got to, if you always are, you're always working on the next best thing. But if you don't take something to clinic, you never go to clinic. There's always something better in the pipeline. But you got to start somewhere and go somewhere. So this is first generation and it's positive. Man, so much better than anything Fulcrum had. Love it. And I'm looking forward to seeing, I hope they show the gene expression data. I'll feel more comfortable to see that the DUX4 really is down. Then I'll say, you know what, over the course of 18 months or 2 years, this is going to be even better improvement. Maybe if you split the cohort into how strong or weak that people are when they start on it, you might find that people at a certain level of strength just maintain that strength, where people with a higher degree of strength still affected actually regain it. Or maybe everyone regains it. I don't know. We'll find out. But these would be the things we'd be kind of curious to know. Again, what can you do now? Sign up for clinical trials. But don't get too discouraged. Arrowhead's going to have their data. We're going to have more trials coming. I'm going to talk about a trial in the next podcast, the EpiCRISPR trial. Well, I'll save my comments on that for that one. But more trials are coming. They're all going to need patients. And the devil's in the detail. Better things can come. But it's good to have something that looks pretty good to start with. Looks okay. If you had choice of this or nothing, you'd sure take this. Choice of this versus Fulcrum, you definitely take this. And quite honestly, if I had FSHD or if I were in it, I'd be trying to get into this trial right now. But I'd also want to make sure that I'm aware of all the other trials. Clinicaltrial.gov is the site, and look for sites and see for the other trials that are going to be out there and that are going to be coming. Okay, there's a very exciting time in FSHD. And when I talk to people about their research test results, if people are affected, almost the worst thing I can tell them is they don't have FSHD, right? The good news is if you are affected and we tell you, hey, you know what, you have the epigenetics of FSHD, go ahead and get your confirmed genetic test. The good news is, help is on the way, and here it is, right? It's help is coming, and more help is coming all right. So great job to the whole team at Avidity. I honestly, that's my honest opinion. You guys did a great job. But you know what? You better keep it up, because I think the competition's coming for you, and it's good news for FSHD patients. All right, man. That's what I got for this. I'll be back with telling you about my trip to Asia and the EpiCRISPR trial coming up soon. I seem to make it through okay talking, so I got to run into the lab and do some stuff. But I hope you like the video thing. This is what I look like on a daily basis. Sorry. What can you do? Hey, we're gonna have Jagger take us on home, man. See.
Thank you for listening to the MyFSHD podcast, where we share with you the latest news and information on FSHD as we strive for a cure. Do you have a question for Peter? He'd love to hear from you. You can reach him at peterjones@med.unr.edu. Thank you for listening, and join us again next time for more news and information.